Hello and welcome to Psychology 301 Learning. This is Dr. Williams, and this is the first lecture in our last series of lectures over animal cognition. So let's talk about animal cognition. Usually when we talk about animal cognition, we're referring to the use of some form of neural brain-based representation or model of experience that's used for a basis of action. And it includes things like thinking and reasoning and memory. And the important thing to remember is that this neural representation cannot be observed directly. We just don't have the sophisticated neuroscience to be able to go in and look at a specific thought or a specific memory. And so we have to infer what's going on from behavior. Now, this is an area of psychology of behavioral psychology that overlaps to some degree with cognitive psychology. And if you recall, back in our first lecture, we talked a little bit about the differences between behavioral and cognitive psychology. And cognitive psychologists rely on internal representations as constructs that bridge our gaps in knowledge of brain physiology. So when we talk about constructs and heuristics, they don't actually exist. When we talk about short-term memory, for example, we don't have anything in the brain that an organ or an area that's called short-term memory. Short-term memory is a very sophisticated construct that refers to all of the physiological processes that allow us to keep something active in terms of stimulus control, in terms of quote-unquote memories. So cognitive psychologists, remember, if you recall, cognitive psychology is a metaphorical science, and so it's willing to talk about and attempt to measure things related to these metaphorical constructs and heuristics. The important thing that you need to know is that these constructs and heuristics are like short-term memory and long-term memory, are believed to encode various types of information. And it's assumed that behavior is guided by these representations rather than by our immediate surroundings when we talk about memory, for example. So we know, for just as an example, we know that the hippocampus is an important part of the brain as it relates to memory. But the hippocampus doesn't work alone. So cognitive psychologists talk about short-term and long-term memory as ways of sort of bypassing the gaps in our knowledge in brain physiology. So a behaviorist, on the other hand, would draw lines around what they would be willing to use as causes of behavior and outcomes of behavior. So back in 1984, Herb Terrace said, the rationale for studying cognitive processes in animals requires no reference to animal consciousness. Now, some behaviorists refer to this field of animal cognition, and particularly the memory area of animal cognition, as the study of long-term or delayed stimulus control, rather than calling it memory because they want to call it what it really is. There isn't actually a thing in your head called a memory. That's an epiphenomenon, and so we prefer to call it long-term stimulus control. So let's talk a little bit about long-term stimulus control and about memory as part of the animal cognition point of view. All right, so animal memory, the existence of memory in animals is identified by the fact that their current behavior, whatever they're doing now, can be predicted by some aspect of their earlier experiences that's no longer present. So performance is controlled by an SD or an S plus that's no longer there. And so the animal, the subject, has to remain sensitive to a stimulus after it's been removed. So let me give you an example. If I show a pigeon a red key light and it pecks at the red key light and it goes away, and later at some point I give it a choice between a red key light and a green key light, if the correct answer is to pick what they saw previously, then they would get a reinforcer for picking red and they would get nothing for picking green. And we'll come back to this procedure later. But the idea is being able to use that previous experience to guide our behavior after that experience is no longer present is what we really are referring to when we talk about memory, whether it's non-human animal or human animal. When you study for an exam, you are presented with the information in front of you, you have all of that content, and then when you go to take the exam, that information is no longer present, and you have to respond using some reference back to that earlier previous experience. All right, and there are a lot of ways to study memory. The couple that I'm going to talk about, I want to talk a little bit about radial arm mazes, I want to talk a little bit about the Morris water maze, and then we're going to go into a lot of detail on the delayed matching to sample procedure. So this is a radial arm maze, and here's a rat foraging on that maze. This actually comes from your textbook. And each arm of the maze, this is a 8-arm radial arm maze, and the end of each arm has a little bit of food, and the rat is placed in the center, and then there's usually a camera mounted above, and they record where the rat goes in terms of foraging. So he goes down this first arm and he gets food here, and to get to the other food he has to go back to the center, and usually this food is in a little cup so it's not visible, and so he has to then decide which arm he's going to visit. And so he has a choice of 7 arms that still have food in them and 1 that does not. And so then he goes down another arm. Say he goes down this arm and he gets food and then comes back. As he visits each arm, he is challenged with having fewer and fewer choices, but those choices rely heavily on his ability to remember which arms he's already visited. So what we find is that the correct number of responses tends to, this is blocks of trials, so this is the mean number of correct responses that rats made in the first 8 choices during blocks of 5 test trials. So this is 5 trials, 10 trials, 15 trials, and 20 trials. And what you find is in the first 5 trials, they only get about 6 right before they make an error. And as they continue with this experience, they get better and better until they're up from a little under 7 to nearly 7.5, so they show an improvement over time. Here is a, what is this? This is a 1, 2, 3, 4, 5, 6. I think this is a 12-arm maze, and you can see it has a little dish at each end so that when the rat is on rat level, he can look down the aisle. He can see the dish, but he can't see what's in it. And so this is 12. The biggest one that I've ever heard of had 100 arms. I think it'd be really cool to do one that was multi-dimensional, so more than one level. So remembering, have you been up, have you gone down, which ones have you visited, which ones have you not? That would be really cool. All right, this on the other hand is a Morris water maze, and a Morris water maze is basically a tank of water that has been something added to it to make the water opaque so that the animal in it can't see the bottom. And there's a platform in the tank and a start location. And so the rat is put into the start location and has to swim until he finds the platform. And a camera above records the path that he takes. And this is an example of, oh, I don't know what that is. Let's get rid of that. Sorry. The camera above on the first, this is our first trial. You can see that he kind of travels around, and here he hasn't even found the platform yet. And so it literally is trial and error, but they repeat this several times, usually once a day or several times, once in the morning and once in the afternoon. And here by the 8th trial, the rat has learned to swim directly from the start point to the platform. So here is a graph of data from a Morris water maze study, and this is the time that, so this is our escape latency, how long it takes them from the time they're put in the water until they find the platform and crawl out. And here it's a function of days of training. So here each day of training, you see that on day 1 it takes nearly 35 seconds. On day 2, it drops to about 12 seconds, and it continues dropping until around day 5. You see that it's only taking about 5 seconds for the rat to swim directly to the platform. So those are 2 ways that we can study memory. And before we move on, we probably should talk, and now's a good time to talk about some vocabulary. So there are 3 important terms that I want to talk a little bit about. The first one is acquisition. Now, acquisition is referred to generally as the early stages of learning. But in memory research, it refers specifically to the period of time during which the individual is exposed to the event or the information that's going to be needed later. So they'll need to remember this information. The second phase is the retention interval. And the retention interval is the time after the event or the stimulus is no longer present before the individual can make a response. So that could be very short or it could be very long. And we often will manipulate the retention interval to study how robust a particular memory is. And then finally we have retrieval, which is the process of reactivating that memory at the time that it's needed in order to be able to respond so that it can guide behavior. So and most of our memory studies will involve these 3 processes in this order. So there's the acquisition phase, there's the retention interval, and then retrieval. And during retrieval, this is usually a test. So let me back up here just for a second. For the Morris water maze, remember that on day 1, we're recording how long it takes him to find his way to the platform. And on day 2, he needs to remember information about yesterday's test in order to improve. And we see that through the use of memory. So acquisition is experience 1. Retention interval is from the first day to the second day, or the second day to the third day. And retrieval occurs when you go back in and on the next day. Here, for example, day 8, they put you in the start location, and you're using all of your experiences from the previous trials in order to perform on the 8th trial. Now, so in a little bit more detail, these mechanisms of memory testing. So how is it that we do this? Well, we typically, in an experiment, a memory experiment, we would present a stimulus or an experience. We would give them a Q, an S+, and we're going to require the subject withhold their response or the test during some retention interval. And then after the delay, we then test for the correct response. In the absence of the S+, we ask them to do something. And if the animal can demonstrate some degree of stimulus control by that original stimulus following the retention interval, then we assume that the subject has retained some aspect of that stimulus in quote-unquote memory. In other words, the brain has is remaining sensitive in some way to that stimulus in order for them to perform above chance levels, whatever chance would be. So the classic procedure involves these 3 things that are directly correlated to acquisition, retention interval, and retrieval. Acquisition, we're going to be dealing with delayed stimulus control, so we really want them to remain sensitive to this stimulus after a delay. There'll be a retention interval after the S+ is delivered and removed, and they're not allowed to respond. And then we give them an opportunity to respond, and we measure performance accuracy in some way. And it's assumed that in the absence of further exposure to that stimulus, that they have to rely on some kind of internal neural representation, however the brain does it, of that stimulus in order to respond correctly. Okay, so now memory has been classified in a lot of different ways. If you think back to intro psych or to your cognitive psychology class, you will remember that we talk about sensory memory, short-term memory, long-term memory as one way of talking about memory. We also talk about semantic and procedural, maybe episodic memory. We also talk about explicit and implicit memory, what you know that you remember and what you don't know that you remember, but you show evidence of memory. Within animal learning, the most common distinction that we use is the distinction between what we call working memory and reference memory. And so I want to talk a little bit about what those are. Reference memory refers to the basic rules of whatever the procedure is, so kind of like the rules of the game. Everything that comes in, all the incoming information, is used with reference to those rules. So just like the rules of poker, reference memory would be what the rules are, and working memory would be the information related to the current game in progress or the current task in progress. So that would be the current hand that's being played. So reference memory really refers to long-term retention of contextual or procedural information that's needed to deal with other incoming information, whether we're talking about a hand of poker or whether we're talking about how to ride a bicycle. It can be, it's long-lasting, it's enduring, the basic skills for how to go about this task. It's difficult to disrupt once it's been established. Even after very long delays, it comes back very quickly. And it's primarily used to organize more specific information. So when we have information coming into working memory, we use our reference memory in order to organize and use that working memory information. So things like the rules of poker or how to shop at Safeway, knowing where certain things are in each aisle, knowing where knowing that the vegetables are at one end of the store and the bakery is at the other end of the store, knowing where the bread is, knowing where the cleaning supplies are, knowing where the shampoo is, knowing where the tortillas are. So knowing where they are is different from what's on your list. So it's really having that organizational information that's why we call it reference memory, because on any given trial, we take the important information and use this, we reference this long-term information in order to successfully use the information we need on this particular trial. So things like how to take exams in my class. It's one thing to remember what's in chapter 10. It's another thing to know how to go about studying and how to take the exams and how to take the test and use the information that's coming in. So knowing what to do, where, and when. Okay. Working memory, on the other hand, is really only retained just long enough to complete a task. So sometimes the things that we do get into more long-term memory. We remember the way we completed an assignment. But in terms of ongoing trial-based performance in the laboratory, we talk about working memory as having information for this particular trial or for this particular task. And after that, it's not necessarily useful. We then go on to the next trial. And so it's short, it's fleeting, it's very susceptible to decay, to loss, and to interference unless you do something to keep it active and get it from short-term into long-term memory. So it's really only useful in the context of more enduring knowledge that comes from reference memory. So for example, you know, the actual cards being played in a hand of poker, once that hand has already been played and a new deck comes out, it no longer matters what you did previously. But while you're playing that hand, you know what's in your hand, you know what other people are doing, and you want to make sure that you play with reference to the rules of poker so that you know how to play. So there's that what's in your hand, it would be working memory, and how to play poker would be your reference memory. What are the items on this week's shopping list? So once you're given a shopping list, taking those to Safeway and knowing how to shop and knowing where those items are without having to look at every item on every shelf, on every aisle, all the way down. So having that reference memory helps make that shopping trip doable and short and brief and successful. The specific content of Unit 5. So we're now working on Unit 5, and that specific content is is useful, but only in the context of understanding how to study and how to take exams for my class. Now, I want to go back and do a little bit of history here. Wallace Hunter was a psychologist, I believe he was in Oklahoma, back in 1913. And, excuse me, he wanted to know whether animals could retain an image of a stimulus. So this is back in 1913, and at the time, Wundt and introspection and personal experience is very popular. Freud is very popular, and Thorndike is one of the few people who's done any research in the area of sort of animal behavior in the laboratory kind of stuff. And so Wallace Hunter wanted to know whether humans could retain, non-human animals could retain an image. So he used dogs, and he used rats, and he used raccoons. And he built an apparatus that consisted of a start box with 3 compartments. So I'm going to describe this. There's a list here. There's a goal box that has a light mounted above it. And there are 3 of them. So, and the light goes on above the one that has the food in it. And so only one of them is baited with food, and it's signaled. And then the light's turned off, and the animal's allowed to guess. So after they learn to discriminate the rules, if you will, the reference memory of go to the light in order to get food, he then can start doing experiments. So here is the here's a diagram of Wallace Hunter's apparatus. So it started with a start box, and in this case the dog is put into the start box, and there's a barrier. The dog can see through the barrier. There are 3 goal boxes. There's food is loaded into one of them, and a light goes on. And so this is the way of letting the dog know the food's in here. And then the light goes off, and the dog waits and waits and waits. And there's he used different delays. And the big test question is, which door has the food? And the goal here I got this weird slide number thing too here, sorry. And so the dog is supposed to remember to go to the light. Okay, so pretty straightforward. The apparatus is smaller for rats, and actually I think he used almost a paddock for dogs. So here are his results. The maximum delay where they were able to demonstrate above chance performance. So in each case, because there's 3 choices, 33% would be guessing. So if he only gets it right 33% of the time, the dog is guessing. Anything above that would imply that the dog is able to retain some neural representation of where this food is located. And what he found was that for rats, that was about 10 seconds. After 10 seconds, they pretty much dropped to chance levels. Raccoons could go as long as 25 seconds, and dogs were able to go for 5 minutes. So, which is pretty impressive. However, he also, in his notes, writes that the dogs and the rats basically cheated. They used body postures to bridge that delay. So, let me come back here. So when the light's on, the dog would move to the side of the box that is correlated with the light. So the dog here would move to the left and stand and put its shoulder, its body up against the left-hand wall of the start box. And if it was on the right, he would move over to the right side of the box. And if it was in the center, the dog would stay in the middle or even lay down in the middle of the box and wait until the door was opened. And rats did the same thing. They used body postures. So they sort of artificially inflated the maximum delay by using these cheating methods, these body postures to bridge that delay. But he also noted that raccoons did not use body postures. They moved around, they investigated, they moved around a lot in the boxes. And so he believed that the 25 seconds that they were able to perform above chance was a good indication of the duration of the memory of a raccoon. So there are a lot of problems with Hunter's procedure, but there's also good things. First of all, this procedure really highlights the distinction between working memory and reference memory. So here, reference memory is the dog's learning that the light signals food. That relationship is part of their reference memory. And working memory deals with where was the light on this trial. And so that really helps kind of make that distinction for you. But there were a lot of flaws in Hunter's procedure. They were very simplistic, and there were a lot of problems. But they paved the way for a much better version, a more sophisticated version of his procedure called the delayed matching to sample procedure. I'm going to stop here. That's a good place to stop. That's 25 minutes is long enough, and I'll meet you back here for our next lecture. Have a great day.